So after DSEG2, we already have a list of genes that's that's overexpressed in either of these conditions. So now we want to look into the gene list and to see which of these biological pathways are these genes actually in. So there are two ways. The David takes the selected list of differentially expressed genes and sees if there are genes in the certain gene sets that are overexpressed, where GC takes all the genes and use the test statistics. So for both of these, we need to have a good definition of the gene sets, and David has its own gene set definition. GC, we need to provide one by ourselves. So as MSIGDB will be a good one that's being commonly used to provide to GC. So we're going to do a very small tutorial on the David. So first, you already have a list of genes that's differentially expressed, like here, and then you click on choose file and this list of genes here, and then you select the identifier, which is this original one. You don't need to do anything on it, and you click on gene set and you submit the list. You submit the list and then you put Homo sapien, which is this, so 9606 Homo sapien, and you submit to the conversion tool. It's going to take some time for conversion. This is the one that I already converted, so I just select this converted gene list and select the specimen, and we do the functional annotation, and this is the result we get from it. So basically, after this annotation, you can after this cluster annotation, you can just download this file. You can analyze on other platforms. On the other hand, GC is a list of differential expressions. Is sometimes so long that is interpretable. Interpretation becomes cumbersome, so and time-consuming. A common downstream procedure is use the gene set testing. So instead of using the each of the genes, we're taking only look of the gene sets. So it aims to understand which pathway and gene networks the differential expressions are implicated in. So in the GC analysis, we need to rank all the genes in the data sets, like based on their log4 change. Maybe you have a cutoff for the p-value, and then based on the log4 change, you rank the genes, and then identify the rank position of all the members of the gene sets in the ranked data sets. You're calculating the enrichment score and represents the difference between the observed ranking and that which would be expected, assuming random rank distribution. So yeah, here you have all these genes and you rank the genes based on this log4 change. You can also have rank the genes based on like a mass formula does not only include log4 change, but also includes the p-adjusted values. So you do the bar plot of these genes, and you can see based on the ranks. So the first couple ones are the ones that's more have a high rank, and the last ones are the ones that's with lower ranks. And then you pass in this ranked gene sets, and you will get something like this. So this basically tells you these hallmarks of oxidative phosphorylation is enriched in this condition. And then you're able to plot to do a plot shows the running yes for the essential score for the gene sets. So the middle point of the plot shows where the member of the gene sets appears in the ranked list of genes. The leading edge subsets of the gene set is the subset of the members that contribute most to the essential score, enrichment score. And for a positive enrichment score, the leading edge subset is the set of members that appears in the rank list prior to the peak score, which is these parts. And for the negative enrichment score, enrichment score, it is the set of the members that appears subsequent to the peak score. So if this is like down here, then it will be this ones. This ones that's been that's the set of the set of the gene members that's contributes to the enrichment score. The bottom portion of this plot shows the value of the ranking matrix as you move down the list of the rank genes. The ranking matrix measures a gene's correlation with phenotype. So the value of the ranking matrix goes from the positive to negative as you move down the rank list. A positive value indicates correlation with the first phenotype, and the negative value indicates the correlation with the second phenotype. For continuous phenotypes, a positive value indicates correlation with the phenotype profiles, and negative values indicates no correlation or inverse correlation with the profiles. So this is like a way to read and to analyze your data and to see how this plot is really talking about. Yeah, so basically these are the materials for this lab. If there's any questions, I'm happy to answer any question regarding to the homework or to the class material. Thank you.